The title of this presentation is RNA Toxicity and Thermodynamics Around Equilibrium, a statistical physics approach for biological systems. To begin with the presentation overview, we'll be describing some connections between heuristic arguments that can be used to describe ensemble level behavior of configurations in statistical mechanics, which also appear in various themes and aspects of systems from synthetic biology, which are subject to problematic conditions such as including toxicity, off-target effects, and other forms of contamination. And to motivate the biological side of our discussion, we will discuss various physics-based aspects of CRISPR-Cas nucleases, which have many variants that can target either DNA or RNA targets in either from a genome or a host organism of interest. And our goal will be that is from a more theoretical perspective, we can describe how some more mathematically related and inclined notions relating to discrete holomorphicity and parafermionic observables in Cauchy-Riemann equations, which appear in complex analysis, break down away from the critical temperature, and how this very much has connections with different type of perturbations that we might be on the lookout for, or try to think about as a computational biologist or as someone who makes use of either genomics or related types of algorithms. And altogether, this will allow us to establish the role between the temperature parameter in more theoretical and applied studies for models with thermodynamic characteristics, which very much fall under many applications and then in the umbrella of statistical mechanics and statistical physics. So the main reference paper that I'll want to begin with is making use of some material that we've also discussed in another circumstance on the channel, which has a thermodynamic characteristic from the COVID-19 crystal structure because it affects the temperatures at which the virus operates and functions at for initiating DNA or genome replication events, which can lead to infection of host organisms. So under the archive, under this perspective from quantitative biology or computational biology in biomolecules, we can study the impact and the probability of obtaining a favorable interaction, which would initiate some virus replication event amongst different binding pockets, which are located on the surface of the COVID-19 receptor. And having this type of information about the receptor structure from the crystal structure is very helpful because if we also want to look at many other broader classes of disease receptors rather than just the isolated incident for the COVID-19 disease receptor, we can also be able to make use of either deep learning or other machine learning algorithms off the shelf for performing some type of supervised optimization problem because we're interested in determining how or which conformation would bind the most strongly to some target of interest. And this is the COVID-19 crystal structure, as we have referred to, in which if we perform a counterclockwise rotation of the crystal structure that's shown on the screen, we can look at the distribution of binding pockets amongst the surface of the receptor. And as in another presentation on the channel, I discuss how there can be connections with the production of protease and how protease inhibitors are related to mechanisms in which this virus propagates its infection. But from a different perspective, we can also make use of this type of crystal structure and this type of interactions that we obtain from binding events, because it's going to give us information about statistical physics and how making use of this more biological setting and describing it heuristically can be used to relate to aspects of a rigorous mathematical argument that I had studied in another case for parafermionic observables, which is very much like a type of theoretically oriented observable besides the types of observables or measurements that a biologist or other experimentally minded people would gather from tuning their parameters very precisely for looking at the performance of a biological system within a very narrow range of temperatures. And the structural information from the protein data bank in the case of the COVID receptor, and also in the case of many other publicly deposited compounds that are located on that website, is that it gives us information about the crystal structure and who was depositing it from the entry authors, as well as the changes from the initial deposition of the crystal structure on the protein data bank, as well as any possible modifications to the surface of the structure 3 years later in 2023. And we can explore conformations that share connections with with the ones below, because these all exhibit different types of compounds for other receptors of interest, and they exhibit countably many conformations that can be produced from either deep learning or machine learning output, which is used to predict, given some force field, which we use to stabilize the molecule within some binding region, how the interaction for the binding affinity would be, or what would be the major contributions, which would determine the orientation of some molecule within the binding pocket, as well as differences representing other combinatorial options for the optimization of the machine learning and deep learning algorithms that are used to produce the conformations, which we can examine manually further to determine which conformation is the best. But despite potential benefits, vaccine candidates can impact undesirable perturbations on the genome or other systems within a host organism. And this is very much related to the overall topic and direction of this presentation, and just highlighting a more narrow range of expected behaviors or expected or undesirable behaviors of biological systems and impacting toxicity to RNA, or even impacting the toxicity to reverse replication, because it very much relates to also the struggles of making use of in silico algorithms and in silico workflows for looking at how we would expect certain compounds to interact that are associated with disease receptors, or different parts of how a disease or how a virus propagates its genetic information. And this is very much related also to synthetic biology, because from a colleague, David Speck, in the Lambert Lab a while ago, we were looking at several other types of directions of research that could be associated with this type of workflow, in which he devised a workflow for measuring DCAS, nucleus dead fitness change after binding effects, with his primary objective being to determine whether the presence of new genetic material can be amplified after a virus propagates mutations to a host genome after infection. So this can very much be related to looking at potentially introducing some type of treatment that can be used from a targeting of a CRISPR-Cas nucleus, like many genomics types of related therapies have been administered. But as potentially as one of the world's most expensive therapies, it's very difficult to determine what would be the secondary lower order effects, which David hypothesized can be related to uh, interruptions to reverse transcription, as well as potentially negatively affecting other parts of the DNA replication process. So it's not very much a stretch at all, or a leap of the imagination, to imagine that there can be other undesirable effects, such as like we have been mentioning and alluding to with RNA toxicity. And the first step of his workflow began with the fact that he chose some viral genome of interest, and he performed some genome mining, which allowed him to enumerate some combinatorial target gene space, from which we can be able to measure through various sophisticated assays that he designed the DCAS, or the nucleus dead binding affinity activity, through next generation sequencing. And having algorithms such as genome prediction or transcriptome assembly can be very helpful for next generation sequencing, because it can help us broaden our repository and our repertoire of sequences, so that we can look at different possible training data sets for either machine learning or deep learning algorithms that we can feed and deploy to these algorithms to determine what would be the type of classification error, and whether, as I've illustrated on some other types of numerical experiments from videos on the channel, whether there be some other type of expected outcome, or like, lack of performance or lack of accuracy of a machine learning or a deep learning algorithm, depending upon the training set that we feed to the algorithm. So this is always going to be very much an interesting direction of research for people in these computationally, in these neighboring computational fields, because they're interested in looking at how you can change the format or different types of information, or the complexity of the format for the training set data, and how this would impact the runtime complexity, and hopefully still being able to obtain some family of algorithms that has a very low classification error, or a low classification loss. And as you iterate maybe more complicated training set data, or look to even go the other way and decrease the complexity of the training set data, how would the type of algorithm and its performance fare? And this is related very much to the to the second item of David's workflow, because he was identifying editing target sites for decast nucleases depending upon whether the sequence was pan-proximal or pan-distal, meaning whether there's some the PAM sequence stands for protospace or adjacent motif, and whether this sequence would either be related more to whether it would either be placed further up or downstream from the target site that we're targeting with the CRISPR-Cas nuclease. And this is also like a numerical type of exercise, because we can look at having some solution approximation for the first passage time, which is generating, some time in which the CRISPR-Cas nucleus, which when it's examining some base pairs of the target sequence of an associated gene of interest, would then be able to pass through all of the energy barriers associated with each base pair of the sequence. And it's very much like related to a first or last passage percolation question, because you assign some type of weights to each one of the edges over, either a configuration for some probability models that you can look over the square lattice, or really any type of lattice that you would like. And then you can look at what would be the time or the optimization problem for either minimizing or maximizing the total amount of time given each one of the weights. So it's very much like a percolation type of question, but it has a biological component because you're targeting the genes, and there has to be a certain type of temperature about which favorable interactions would occur. And in this case, under a very mild assumption on the landscape potential that's formulated, as I've discussed in other short conference presentations, which can be given as a second order partial differential equation, we can have some type of analytical solution, which is good for generating some type of preliminary idea of what the passage time distributions would be for the CRISPR-Cas nuclease amongst all the base pairs of the sequence that's expecting. And there can be, as we show on the left here, from the first passage approximation that we can obtain numerically to obtain some list of times, which is used for the solution approximation. We can look at several different types of Cas nucleases, whether it be Cas9, Cas12a, Cas13a, or Cas13b, to name a few. From this 2018 reference. And this gives us some type of more biologically oriented interpretation of the nucleus specific characterizations of binding specificity and editing efficiency, which can very much relate to the types of questions that people in the lab would want to study through some type of assay development and trying to look at oligos or different types of small plasmids for looking at what would be the output associated with different types of genetic circuits, which could be experimentally paired up to certain CRISPR-Cas nucleases, whether they make a single-stranded RNA break or a double-stranded DNA break. And degradation to components of biological systems can occur under many circumstances, such as the one below, in which in the third step of his workflow, David looked at the capability to measure RNA degradation from each combinatorial target, which comes into play from several ingredients from synthetic biology, in which within this plasmid, there's a Cas13a plasmid system, as well as an associated repression mechanism, as well as we had alluded to earlier, a guide sequence which serves to localize a CRISPR-Cas nucleus when it's first performing its edit. And if there's a match sequence or a non-match sequence, if even in the case of a match sequence, there can exist a non-specific RNA degradation which can impact a fitness penalty. So this is exhibiting some types of undesirable side effects and lower order effects, much like some types of notions with relating to observables that will mathematically make precise a little later in this presentation. Because even if we have a perfect match between the sequences at the CRISPR-Cas nucleus and specs from either the DNA or the RNA guide, there can still be a fitness penalty associated with inspection of the site alone. It's kind of like in quantum mechanics, how even if you observe the particles in a system, you can still have some type of deterministic effect in fixing the positions and the momentum of the particles within this ensemble. And in this biological system, there's kind of this same type of penalty or a very similar type of penalty, because you're already impacting some fitness changes, some undesirable effects through either RNA or DNA toxicity, which, as we had mentioned, can impact thermodynamically reverse replication. And this can also have implications for interruption of DNA replication. And toxicity to RNA can be impaired under other circumstances, which can further impact other functions of the genome, such as those listed in this reference, in which it can occur within a non-coding repeat expansion disorder. And some candidates from the list of possible homology models include that when we're looking to make use of this thermodynamic interpretation, like we have been advancing from statistical physics, we can look at building several possible homology models, which can have implications for how different structures of the compound connect together, which would then influence the types of regions of the compound that we would like to examine further with our algorithms to computationally approximate different conformations which could interact strongly with the site of interest over the receptor. And now we can get to the discrete holomorphicity side of things and associated objects and describe from a bit of a more technical perspective how some of these more general still characteristics arise from the biological characteristics that we started out and how we were motivating that with experimental side of things. And that we want to look at introducing a very specific type of observable, as well as the fact that from some very classical and simple mathematical relations like partial differential equations that even describe master equations and translation of proteins and other bodies within the genome can be used to establish some types of properties of this observable. And we're going to be introducing several works which relate to parafermionic observables for a wide variety of models, the Cauchy-Riemann equation, critical behavior and how this relates to thermodynamics, irreducible representations, and several other connections, whether it be related to algebraic connections or even divergence of the correlation length, which even has a biological type of consequence and interpretation because we're positioning certain types of agents within the genome or the transcriptome to interact within very small, appropriately defined biological systems, as well as connections with vertex models. And this is given by the fact that there's some notion of massive S holomorphicity for the Ising model, which is a classical model of statistical mechanics, which I'm not going to define in this presentation. It's just used to model ferromagnetism. And is given by the fact that for some of the definitions of in the perspectives that we encountered in a more physically oriented type of system can be realized from the properties that unobservable that mathematicians and physicists look at, because from the more applied perspective, we're looking to study the behavior of our ensemble of different configurations. And in this case, for the nucleases, we were looking at how they can interact with certain binding sites or sites of the genome or from either DNA or RNA guides that want to target. But this observable is very much satisfying its own type of conservation because we're looking at a conservation type of law from the observable depending upon all degrees of freedom. And whereas some nucleus can just have a degree of freedom amongst moving either upstream or downstream from some target strain of interest. In this case, the parafermionic observable has 4 degrees of freedom, which can be given by looking at either fixing one direction and then looking at multiplying the orientation of the observable amongst an opposing direction, which could then be related to, as is given in the first equation from the system of relations, the value of the observable amongst the edge E and the opposing edge to the east edge. So this is to say that the interpretation of a thermodynamic interaction in temperature, it comes into play in some very simple set of relations like given from these 4 relations, which we can look at for studying about every possible direction that the observable can travel. And it's very much relating to some type of evolution or concept of time evolution and how even biological systems they can evolve in time and explore different parts of their landscape. And from the associated penalty that which we also have for different directions that the parafermionic observable is defined on, below this critical temperature, the Ising model, where ferromagnetism, which has properties of ferromagnetism, we can also see that it's almost paralleling a very a less precise notion of a thermodynamic stability, which we encounter in synthetic biology. But there also exists, as we had alluded to, a propagation mechanism for the Ising model, because we can look at defining a propagation mechanism for the Ising model away from the critical temperature, because in this work in which I looked at extending some previous results from a 2012 paper, below the critical temperature, they define some interval, which is like a half interval, the real line. And then you can also define some other type of related interval besides I star, which is given by boldface I. And you can define the propagation mechanism, which is a function, a mapping from the two-dimensional real space into itself, which is given by the fact that we can define the action of this propagation mechanism either on the points K sub L, or K sub R, which are either demonstrating either points along the leftmost boundary or the rightmost boundary of some finite volume, or points within the bulk of the finite volume, which is very much relating to some process of propagation mechanisms and how nucleuses and other biological systems can, within a certain range of temperatures, be able to advance themselves forward in an environment. So it's very much demonstrating how this mathematical object, which we're trying to study from a more rigorous type of perspective, it's very much sharing and paralleling many types of characteristics of what we would expect a system to physically do from experiments and types of assays that we would use to measure its activity. And there can be an equivalence between two models, which it's more of a it's more of a theoretical consequence that we can use because we can establish an equivalence between the high temperature expansion of a loop model and the Ising model, which is related to the big O of 1 measure. And I won't really go too much into what this is related to, but it's just stating from the fact is that if we fix some inverse temperature, which is used to define the Ising model, the interactions of the Ising model and how strongly our magnet can be ferromagnetic, under a certain degeneration of parameters of the probability measure of the loop model, we can establish an equivalence between these two models. And this is to say that it can also, as I'll allude to at the end of the presentation, establish some type of consequence about whether there are any types of helpful characteristics and associations that we can make between other models or physical systems of interest. And as we had just mentioned, this allows us to make associations between many different systems, in which we can look at either choosing some genome of interest, which has all of its intrinsic targets, and looking at the distribution of the coding regions or the non-coding regions. And then this can be related to optimizing some type of function of an enzyme, because as we saw in the case of COVID, it was able to result, unfortunately, in many types of sicknesses and ailments associated with this. So it's really a direction of research to determine how certain characteristics of enzyme design can help us, uh, you know, not really, not really optimize function for, for the cause of an infection, but even other causes, which can be used to maybe optimize the production of biofuels or other more industry related applications, which can ultimately be related to in silico algorithms and how they're trying to approximate the behavior of different conformations within a very narrow and specific range of temperatures. So the concluding remarks that we have is that we introduced components from previous presentations on the channel for in silico workflows in which hundreds of compounds can be screened against one another. And we highlighted the wide variety of biological processes that can occur when receptors and ligands interact, which can share connections with interruption to reverse replication in systems from synthetic biology. And also, as we had mentioned, be able to impart toxicity to either DNA or RNA. And this helped us demonstrate many areas of current interest for applying machine learning pipelines, whether open source or proprietary, for exploring hundreds of new compounds within the enzyme space, and also how there can exist many theoretically oriented objects of interest related to this discussion. Thanks for watching.